Human Genetics, University of Utah. He's focused on investigating the neural circuit and cellular mechanisms underlying the microglia and neuronal interaction in repetitive behaviors using multidisciplinary neuroscience approaches that include genetics, behavioral, optogenetics, miniature fluorescence endoscopy, electrophysiological, and computational approaches. I'm just going to ask him to get started since we're running behind, and you will learn more.
Yeah, thank you very much. Good morning to everyone, and thank you, Pramila, and the entire Brain Foundation for inviting me here for giving this talk. So today I'm going to talk about calcium transients in microglia and how calcium is important for driving repetitive form of autistic behaviors. So repetitive behaviors are evolutionarily conserved behaviors. It's found in invertebrates like Aplysia, C. elegans, and it's also found in Drosophila, zebrafish, and it extends to mouse, non-human primates, as well as in humans. So repetitive behaviors in humans is observed during early development, especially in autism spectrum disorder kids. And this could be either the hand flipping type of behavior or repetitive lining of the objects. And once the kids enter into the teen age, they have a lot of speech regression, and also they exhibit loneliness. And finally, they are socially isolated from the environment as well as from other kids. And this poses a extremely high challenge, not only for the autistic kid brain development, but also for the parents to manage these children. And as the number suggests, there is a high increase in the autism rate, not only within the United States, but also worldwide. So this is my own son, Dave, who is a high-functional autistic, and he was a happy kid earlier, and then he was diagnosed with autism when he was 3 years old, and now he is 12 years old. He exhibits repetitive verbalization, repetitive visual stimuli is what he craves for. He is on repetitive task looping all the time. And he has a lot of sensory issues, like as seen in his school setting, where he was awarded for being the builder of the month. He has to put the headphones all the time, because if he's loaded with more of sensory information, he cannot handle that information. So he's also exhibit the social interaction deficit. He's very good by himself, but when it comes to interacting with new people, or when it comes to interact with the people he's familiar with, he interacts to an extent that he can utter 3 or 4 words, and then the question, and then he stops. He is by himself into one of the repetitive behaviors. So that's what I call social isolation. And he is struggling very much in math as well as comprehension, and he also have problem with cognitive coherence. So we have carried out a lot of therapies like speech therapy, occupational therapy, social therapy, ABA therapy, and it has improved his ability to operate at the level he is operating. And the effects has been working on his language improvement. He's good in W questions like what, where, and why nowadays. And he has improved attention quite significantly. And he's also self-regulating himself and self-balancing himself. And that brought me into the next slide, which is, I wanted to understand in the mouse model if there is any good model to study repetitive behavior. And in my search, one of the criteria that I had was, the model should be good enough so that we have the connection by a single gene which might be working on a specific cell type, acting on a particular neural circuit which might be controlling a particular behavior. And my search actually led to this fantastic paper that came from Dr. Kapike's lab in 2002, where they had created a loss of function mutation in HOXB8 gene, and the behavior turned out to be excessive grooming. And that was a paper that was published in 2002, and it was a very important paper, especially for studying the repetitive behavior. So by 2010, it was clear the cells that were marked in the brain were microglia, and that microglia happened to be a second population of microglia, unlike non-HOXB8 population of microglia. And by 2017, I was able to show that it was corticostriatal circuit on which the HOXB8 microglia was impinging its action, because of which we got the behavior. However, the causality link was missing in the entire story. So by now, everybody accepts that microglia is not only important for repetitive behaviors or autism or addictive type of behaviors, but it also plays a huge role in neuropsychiatric condition like schizophrenia or depression, or even in neurodegenerative condition like ALS, multiple sclerosis or Alzheimer's disease. So it's important to know, what's the function of these microglia, how are they communicating with the neurons, what is its action on the neural circuit, and how they are responding to neural circuit activity and involved in behaviors. So in order to go about in understanding this question, I asked this fundamental question. So how does HOXB8 microglia respond to neural circuit activity and behavioral changes? And why neural circuit specifically? Because we understand that neural circuit process information via synapses, and synapses are fundamental unit of brain. So hopefully, if we know how neural circuit communicates with HOXB8 microglia or the other way around, we would probably be able to understand how they're operating at the mechanistic level to induce a specific behavioral output, and in this situation, it's repetitive behavior, microglia. And indeed, the first few attempts, as with any other experiments, it's very challenging, because we are not studying neurons, which is 50 micron in diameter, but we are studying microglia, which is 10 to 15 micron in diameter. So the challenge was to put a mini scope on the head of the mouse and allow the mouse to behave and measure the free calcium in HOXB8 microglia alone. And the way how we are measuring is by using a calcium sensor called GCAM5, which is specifically present only in HOXB8 microglia and not in any other cells. And that is shown in the striatum in this situation. And here is an implantation of lens, green lens, which is 4 millimeter in the length and 1 millimeter in diameter. And it is targeted to striatum. And the experiment is very simple. We allow the mouse to behave normally, and then we sprinkle a little bit of water on the head of the mouse in a control mouse. And in response to microsprinkle, you can induce phase 1, phase 2, phase 3, phase 4 type of behavior associated with the repetitive grooming. And the question is, what happens to HOXB8 microglia calcium in response to the behavioral induction? So shown here is one of the mouse subject which was implanted in striatum. And in response to the behavioral induction, I was able to observe a rise in calcium in HOXB8 microglia. And each of these colors represents individual microglia. And these are just the heat maps showing the increase in the calcium levels in response to the induction of grooming. And once the grooming stops, the calcium levels also comes down. And this was repeated in multiple subjects as well as multiple areas of the brain. And in here, I'm showing just the dorsomedial striatum. However, if you create a region of interest in a location which does not contain microglia, you just see the oscillation, but you never get a real calcium transient with a clear onset kinetics as well as the offset kinetics. So this data was reproduced in multiple subjects, and here shown is subject 1 and subject 2, and you can see the increase in the calcium level in all of these microglia population. And it's variable, and the variability comes from the location of HOXB8 microglia as well as the vicinity of HOXB8 microglia to the particular circuit which is getting activated in order to induce the repetitive behavior. So shown here is a correlation plot to determine the activation and co-activation, where during the pre-stimulation of the behavior, you don't get the calcium signal, but in response to the stimulation, you get the increase in the calcium level, and during post-grooming behavior, the calcium goes down. And this is just showing the correlation plot for the pre-grooming, during grooming, and post-grooming, suggesting that all microglia are co-activated at one time in response to the induction of the behavior. So I repeated this experiment in a second region, medial prefrontal cortex, which is also part of the corticostriatal circuit, which drives the grooming behavior. And even in this situation, I was able to elicit the calcium transients in HOXB8 microglia, now using a GCAM6F, which is another calcium sensor marker. However, if we do a stimulus-free natural behavior, you don't get those calcium transients, which suggests that the calcium transients are in response to the induction of the repetitive grooming behavior. So shown here is comparison of session 1 and session 2 from the same mouse that was recorded. And as you see, the activation of behavior induces the calcium transient, but you can also get multiple bouts of calcium transient based on the number of bouts of grooming behavior the animal is exposed to. So shown here are the calcium transients of multiple bouts. And at that point, we thought probably we need a much longer amount of grooming behavior in order to record calcium transient for much longer time. So in order to do that, I came up with a simple idea of dropping a small oil drop on the surface of the skin of the mouse, which induced a lot of grooming behavior. And in response to the induction of those grooming behavior, we got beautiful calcium transients in multiple microglia. You would appreciate that some microglia here show a lot of activity, but some microglia show very minimal activity. And again, the reason why we might have observed these kind of differential calcium responsiveness of these microglia is probably attributed to how close they are to the circuit versus how far they are from the circuit. And you can get different shapes of the transients in these microglia. And as you see in multiple session, in a high magnification, the calcium transient response are variable across different sessions, from the same mouse, from the same area of the brain. So shown here are just the heat maps as well as the correlation coefficient of the same data that I showed you, where the low activity cells compared to high activity cells was significantly lower. The fast onset or fast offset of the response for the high activity as well as low activity was not significantly altered. The total events were more or less the same, no significant change. And the total events also was not significantly altered. So shown here is the histological verification of the HOXB8 microglia expressing GCAM5. And these tiny little stars are the HOXB8 microglia that we are visualizing in the dorsomedial striatum. So, one question is, if you go to a different part of the brain, such as somatosensory cortex or basal cortex, and stimulate viscous, do you see similar kind of calcium transient? And the answer was no. It was simply the oscillatory type of activity that was recorded in this situation in response to viscous stimulation. However, the calcium transient was very specific to the repetitive behavior as well as the anxiety behavior that I tested in a different experimental setting. So the last question was, what is the nature of calcium transient in HOXB8 mutants? Because HOXB8 mutant exhibit excessive hair loss as well as excessive repetitive behavior. So the question is, how does calcium transient looks like? So to do that, I used this mouse that was created in Mario's lab, HOXB8 XIRS3, and crossed that to GCAM6 that was carrying a HOXB8 LOX, and got the lens into the mutant mice and investigated what happens in those mice. So unexpectedly, the calcium transient were completely absent in the HOXB8 mutant microglia. I wasn't able to see any calcium transient whatsoever in the HOXB8 mutant microglia when the animal was completely engaged in the grooming behavior. And this was a little puzzling for us because all what it was suggested was a failure in the ability of regulating calcium in order to control the behavior. And that failure probably was leading towards the repetitive grooming behavior. So shown here is a movie, I call that as time t equals 0, where the calcium is always elevated in the mutant brain and it never comes down, which is what leads to the failure of the microglia to regulate the calcium. And even after you come for 30 minutes, and if you run the same experiment, the calcium levels in all these microglia will remain the same. Even if you come after a week, 2 week, 3 week, it's the same. The calcium will not go down. And that's very problematic for the brain. Had it been neurons, probably the mouse would have died, because we all know how calcium dysregulation is detrimental to the nervous system. So, of course, one also has to ask, when the behavior is induced, is there any change in the neural activity? And in order to demonstrate that, I looked into CFOS staining, which is a early gene expression marker in the neurons, and noted that in response to the behavior, the implanted site had higher expression of CFOS. However, the spared hemisphere did not show any CFOS increase in the expression. And that's what is quantified here, which showed a significant increase in the implanted versus spared side, although the microglia on both sides were pretty much the same. So interestingly, when the behavior is generated and when microglia are eliciting the calcium transient, we also detected increase in the Kv1.3, which is a membrane depolarization marker. And Kv1.3 increases, and that is shown in prefrontal cortex as well as in dorsomedial striatum. And we have tons of data that shows increase in the Kv1.3. So, however, if you look at the control conditions where there is no behavioral induction, you won't get any increase in the Kv1.3. So that suggests that Kv1.3 increase is also related to the activation of microglia as well as the increase in the calcium levels. And that's what is quantified here. And here is a model that I put to explain the experimental outcomes. We think that HOXB8 microglia and non-HOXB8 microglia works on this mechanism, the brake accelerator mechanism. By creating genetic mutation or by creating behavioral manipulation, you perturb the HOXB8 microglia function, and in this case, the calcium regulation, and that is what is leading towards repetitive grooming or anxiety-like behavior. And summarizing my data, what we found was the HOXB8 microglia elicits calcium transient in response to grooming behavioral induction. Repetitive grooming behavior increases neuronal activity within frontal cortex as well as striatum. Repetitive grooming behavior increases the expression of Kv1.3 on HOXB8 microglia, and HOXB8 mutants show calcium regulation defects in HOXB8 microglia. So what's next? We have to identify what receptors or what signaling pathways that are important for controlling the calcium in microglia, so that we understand more of the direct implication of the calcium receptor or calcium channel, be it on the surface or be it intracellular. So we are not yet there, but one day we'll probably go there. So with that, I would like to acknowledge Dr. Mario Capicchi, who is here, and the Capicchi lab, which has helped me to grow as a scientist, as well as which has given me enormous independence and everything that I wanted to do in terms of experiment and achieve some meaningful experimental outcome after several years. I would like to thank Inscopics, which awarded the Decode Award, which allowed us to buy the cameras, the SEED grant and research instrumentation grant, the Brain Foundation, and thank you very much for your attention.
Thank you very much.